Hello students, good morning. I'm your educator Dolce, and as you all know, we are on chapter number 3rd, that is human reproduction of unit 1st. So as we have done the male and female reproductive system in the previous few lectures, today I'm going to start up with the process of gametogenesis. So gametogenesis is the topic that is very important from your NEET and theoretical examination point of view, and you will definitely get 2 or 3 questions from this topic. Okay, so let us start that first of all what the gametogenesis is. So the production of gametes from the germinal epithelium of the gonads is known as gametogenesis. Gameto matlab gametes, genesis means generation. So the process of generation of gametes in the gonads of males and females, that are testes and ovaries respectively, is known as gametogenesis. Gametogenesis is a kind of process in which reduction of the ploidy of DNA occurs. As you know, gametes have the half the content of DNA incomparable with the human or the animal body. It means they undergo the meiotic division. As I told you in class 11th cell division chapter, that meiotic division is the process in which the reduction of genetic material occurs up to half. So, for necessarily the reduction of genetic material, the replication of DNA occurred and it resulted in formation of 4 gametes. Out of those, the 4 gametes of the male are functional, while that of female only 1 is functional out of those 4 formed. Gametogenesis is generally of 2 types, oogenesis and spermatogenesis. So firstly, I am going to explain you the spermatogenesis. As I told you in the previous lecture that spermatogenesis occur in the seminiferous tubules of the human male testis. As is shown on your slide, that seminiferous tubules are tube-like structures present in the testes, and they are the functional tissue of the testis to produce gametes. Okay, so as is shown in the slide form, as also in the form of chart, and on the right side of the screen you will are able to see the actual transformation of the germ cell into the spermatozoon. So firstly I am going to explain you the diagram number 1, that in the testis Leydig cells are present, and on the basement membrane, that is known as the germinal epithelium, the male cells are present, that are said to be the spermatogonium. They are diploid cells, and gonium stands for they have tendency to generate the gametes. Okay, so the spermatogonium in fusion with the Sertoli cell by harvesting nutrition from the testes, they undergo some time of division to produce the stages. Firstly, it forms the preleptotine spermatocyte stage, then it produces the pachytine spermatocyte stage. And I hope you memorize all these stages of meiotic division. These are the divisions of meiotic process or the meiosis of cell division. Okay, then whenever these spermatocytes, primary or secondary, they form, they convert up into the spherical structure or cells. They are said to be the spermatids. As you know, and as I told you, spermatids are non-motile sperms. Actually, they have the capability or the genetic material similar to just the spermatozoon, but the problem here is they are immotile in nature, and according to the process of fertilization, the necessary point is that the sperm should be mobile. So the process which convert the elongating spermatid into the spermatozoon that is mobile, it is known as spermiogenesis. So the whole process of conversion of spermatogonium into spermatozoon, from one spermatogonia 4 sperm cells or spermatozoa are produced. That process, whole process, is known as spermatogenesis, while just the conversion of spermatid, immotile spermatid, into elongated motile spermatozoon, it is known as spermiogenesis. Okay, so please now focus on diagram number 3, that we have primordial germ cells, means primordial means primary germ cells that have diploid DNA content. Okay, they convert up into, or they undergo mitotic division to produce two kind of cells. They are known as spermatogonial cells, type A and B, depending upon. Type B has a tendency to undergo the division, and type A convert back into the primordial germ cells. So the type B cells, that is spermatogonium cells, they are diploid in nature. They undergo mitotic division to produce diploid primary spermatocytes. Okay, up to here the division is mitotic, but the replication of DNA occurs. From this, it undergo meiosis 1. It undergo meiosis 1 to produce secondary spermatocyte. So, from primary spermatocyte, that is diploid, two secondary spermatocytes are produced that are haploid in genetic constitution. From those haploid secondary spermatocytes, by undergoing meiosis second, 4 spermatids are produced. Means from each secondary spermatocyte, 2 spermatids are produced. It is meiosis second. As you know, meiosis 1 is reductional. So the diploid primary spermatocyte convert up into the haploid secondary spermatocyte. While the meiosis second, it is equational, or it is just similar to that of mitotic division. Hence the secondary spermatocyte just convert up into the haploid spermatids, not into the diploid content. Ultimately, these spermatids, they get converted up into the spermatozoans, that is by the process known as spermiogenesis. Okay, so we have primarily 4 stages. First 3 are mitotic, then meiosis 1, then meiosis 2, and ultimately the spermiogenesis. All these stages collectively occur or take place to ensure the process of spermatogenesis. Okay, so what you have to memorize that which stage is responsible for which content of the DNA or ploidy of the DNA, and also the structure of the cell formed during that process of division. Okay, I hope you understand. Now, say this diagram here, the whole process of spermatogenesis, it is represented in the form of phases. Means we mainly study the process of spermatogenesis under 3 phases or 1 conversion. First is multiplication phase, from which the spermatogonial cells, they undergo mitotic division to produce number of primary spermatocytes. Then is the phase of growth phase, in which primary spermatocyte, it undergo meiosis 1 and meiosis 2 to produce spermatids. In maturation phase, just the spermiogenesis occur, and it result in the conversion of spermatids into the spermatozoon. I expect that you get a better idea of the process of spermatogenesis from the following diagrams. Okay, so now we are on oogenesis. So as is shown in the diagram displayed on your screen, oogenesis is just similar to that of the spermatogenesis, but here some kind of special structure are formed that are known as the polar body, which are dissimilar to that of the sperms produced by the male. In male, 4 gametes are produced, as I told you, but in oogenesis only one functional gamete is produced, that is known as ova. So the oogenesis starts with this type of cell that is known as oogonium, which is diploid. Similar to the first one, it is known as multiplicative phase. So it undergo mitotic division to produce additional oogonia. From that, the second phase start, that is known as the growth phase, in which from oogonium diploid primary spermatocyte is produced, and it undergo meiotic divisions. In the maturation phase, they undergo 2 divisions, meiosis 1 and meiosis 2. In meiosis 1, one polar body and one secondary oocyte is produced. Means instead of producing 2 similar gametes, they produce 2 dissimilar bodies. The smaller one is said to be the first polar body, and the larger one is said to be the secondary oocyte. At this stage, ovulation occurs. Means the release of secondary oocyte represents the stage of ovulation, which occur at the 14th day of ovulation or the menstrual cycle, with the peak of LH hormone. Okay, so after this, whenever or sometimes if the female it copulates with the male and has received the sperm, only then the fertilization occur and it produces a functional ovum, mature ovum, that ultimately forms the zygote. Okay, and the first polar body and the secondary oocyte means the first polar body it divides further to produce two more polar bodies, and the secondary oocyte, if it received the sperm, only then it produced one polar body and one mature ovum. Okay, in this way, in this whole process, we harvest three polar bodies. All these three polar bodies degenerate and we are left with only one functional gamete, not with the four as we observed in the spermatogenesis. Hence, in this way, the oogenesis is highly wasteful process. Let me tell you something interesting, that whenever a female baby is in the womb of the mother, it has follicles similar to or expected to have in 10 millions. With the time it get degenerate, and when a female is take birth, the functional ova or the follicles left only 2 to 4 million. Whenever a female get mature, its number reduced to 40,000 only. Okay, at the onset of puberty, the functional follicles in the female ova ovary are only 40,000. Out of those 40,000 follicles, as you know, a female can release only 4 to 450 ovas throughout her life only, because as you know, the pubert life of a female start at the time of 10 to 12 years, and it ends up with the menopause at the age of 45 years. So the total age is just of 35 years. In 1 year they are 12 months, so in 30 years we have approximately 600. Okay, so in those, female may pregnant in 1 year. Okay, so the number of egg reduce. So in this way, the functional ova produced by a female during her reproductive fertile reproductive period are only 450 to 500 ova. So from 10 million, only 450 functional ova are left in the female ovary. Hence, the oogenesis is considered to be highly wasteful process in comparison with the spermatogenesis that we observed in human male. Now come to the second diagram so that we can theoretically get an idea about the process of oogenesis. So the firstly the oogonium, it undergo mitosis during the early fetal life, means when we are in the mother womb, to give rise to primary oocyte. Okay, then those primary oocyte during the fetal development, they undergo or they start to undergo meiosis 1. After puberty, these primary oocyte means the primary oocyte stage is of very long years, means from the birth up to the onset of puberty, means 10 to 14 years. So meiosis 1, it is the stage at which the ova arrest. You may definitely get a question, name the stage at which ova arrest during the fetal development or after the fetal development up to the onset of puberty. That stage is known as meiosis 1. So after puberty, primary oocyte complete the meiosis 1 and produce secondary oocyte and first polar body. As you know, first polar body, it may or may not divide, it is not always necessary. The secondary oocyte, it undergoes meiosis second only when it receives the sperms. Okay? After fertilization, meiosis 2 resumes and the oocyte, it splits up into an ovum and a secondary polar body. So in this way, 3 polar bodies can be produced or 1 polar body can be produced. After fertilization, the nuclei of the sperm, it unites with the ovum to produce a functional diploid zygote. So in this way, the ovulation, it occurs at the day 14th when it receives the sperm. Only then it undergo fertilization and completes the meiosis second. If there will be no sperm, no meiosis second will occur and there will be no fertilization and production of a diploid zygote. So in this way, we have done or completed up the process of gametogenesis both in males and females. In next lecture, we will study about the male and female gamete structure, that is ova, Graafian follicle, and the sperm. In